Can you guys see my screen? Just use the thumbs up button as well if you think you can see me. Oh, okay. Um, so I'm going to make a very brief presentation. Because I've spoken with Paninga, and the goal of this meeting, this summer program, is to get you to look at and see different types of research being conducted at different universities. So what I'm going to do is I'm going to tell you a bit about my research, tell you a bit about myself. I'm going to take questions you might have. It might not be directly related to the research. It might just be something you've had at the back of your mind that you've been wanting to talk about. Just feel free. I think we're going to do like a 20 to 30 minutes presentation, and then we're going to go on to take those questions. So if you have questions, please write them down. And so I'm going to try my very best to make this short and interesting. So I'm going to talk a bit about my background, about what journey I've had. I've looked at, some of you have introduced yourself, and I think some of you, we might have close backgrounds. I think it would be helpful for you to know what I've gone through and how it has helped with the research I'm currently doing. I'm going to give you a brief talk about what I'm doing here at the university, at Michigan State University. I'm also going to talk about how exactly do I carry out my research. And then I've named it an open house, but essentially I'm just going to take your questions. Now, please feel free to ask any questions that might come to mind. No question is a stupid question. If you cannot ask directly, you can just type it in the chat box, and the moderator is going to get the question and they're going to read it out to the hearing of everyone, and then I can answer. But to give you a brief background of myself, which you've talked about already, I studied biochemistry at the University of Nigeria, right? And then I did a bit of teaching during NYSC. And then I stopped, and then I chased money for a bit. I left the entire biochemistry and I worked in the bank. And I also worked for PwC in Nigeria. You can ask me questions on that as well. Feel free if you're confused, or if you think if you think it's interesting. And then here I am, finally, I'm doing research. And so what I really want you to take out of this slide is that you don't need to have like a certain type of background. That you can come from different types of backgrounds and you still end up doing research if you're interested, if you chase it correctly. And I think what the Pani Ward Foundation is doing, I think it's commendable. I have to commend Paninga, and I'm grateful to this opportunity, right? Because I think like myself, when I was in my final year or my third year, I didn't know how exactly I was supposed to pursue research I was interested in. And so the message I'm trying to pass across today is that no matter what you think you've been doing in your background, if you're really interested in doing research, you can finally end up as a researcher. And so this is just like a bit of encouragement. And so the first thing I wanted to talk about is, what exactly is research? I'm sure your session yesterday was nice, and you know what research is, but I'm just going to rephrase it again. I'm just going to talk about it again because I think we need to drum home exactly what exactly is research. Research is everything you do that aims to solve a problem. And so here I have a guy on the screen, and you can see the plate in front of him is empty. And so the question is, what is the problem in this picture? If you take like 30 seconds to think about it, the problem in this picture is hunger. And so how do you solve this problem? You give him food. This is research. Don't forget, like I've said, research is aimed at solving a problem. I'm going to talk about what I'm trying to do here at the university to solve certain problems, right? But I just want you to have it at the back of your mind. So you don't need to do a very big thing. You don't need to discover a new drug, or you don't need to create a new chemical. You don't need to win a Nobel Prize for you to do research. Just know that research is everything aimed at solving a problem. I have also this picture here, and so we can see this little baby crying. And so what is the problem here? The baby is crying. And so what are the activities we use to solve that problem that the baby is facing? You give him a toy and he's happy now. And so, like I said initially, everything that you do that is aimed at solving a problem is research. And so one thing I would like you guys to do is to look around yourself. Maybe you're still an undergraduate, or you're working. Maybe there is no data on the number of sick students in the university, or the number of students with, let's say, malaria, or the number of students with. That is a problem, right? You can start to look at how to solve that problem by maybe printing, collecting data on students that have malaria. So what I just want you guys to do is just look around yourself, try to solve problems. When you get this mindset in, you're becoming a researcher even before you come to do a PhD in the university. And so start now to try to learn how to solve problems. And when you try to come for your PhD, if you come to Michigan State University or any other university you're interested in, it's going to be much easier to carry out scientific research. And so now I'm going to talk a bit about what I'm doing here at the university, how I intend to impact and help humans. And hopefully I'm able to attract some people to apply to Michigan State University and to try to do the type of research I'm doing. I hope I'm able to achieve that. And so I'm going to talk about my research. Don't forget, like I said earlier, research is essentially solving a problem. And so here, what you see is you see a person on the screen, right? Some of you might recognize this person. But in my research, what do I see? This person, I don't see him as a human, just like a human. I'm looking at the fact that this human is built with certain materials, proteins, carbohydrates, lipids, DNA. So essentially, my research tries to understand how exactly do these tiny building blocks help build an organism? How do we help develop an organism, right? And so, do you know when life starts? Life starts from fertilization, where you have a human egg being fertilized by sperm, right? I'm assuming that a lot of us have done introductory biology and then we have an idea of what we're doing, right? But please, if you have any questions, if you think it's a bit vague, please ask your question. So don't forget that the goal of what I'm trying to achieve is to tell you a bit about my research, what I'm trying to do to solve problems that exist in the world, and try to get you interested in doing research in structural biology. So what I'm doing at the lab presently, tell the story. So like I said, life starts first at fertilization. We have a human egg. We have a human egg that's fertilized by sperm, right? And so what happens is that development takes place and the egg becomes a baby, right? And so growth continues to occur. And that baby, after 9 months, the baby is giving birth to, and then we have a child. As the years go by, the child keeps growing, right? And we have a full human adult. But in my research, what I'm trying to understand is how exactly does this occur? How is it that from just a single cell, we have a few human that has hundreds of billions of cells? So what exactly is happening is that we have just one cell, but the cells divide as the organism keeps growing. And as the cells divide, they form organs, they form systems, and you have the full human. But for that to be able to occur, you need cells that are, as the cells are growing, the cells need to be able to stick to each other. So what exactly I'm studying at the university is how exactly do these cells stick to each other? Do we use cement? Do we use super glue? Do we use putty? Do we use clay? How exactly are these cells able to stick together? And why is this important for us? Is because when this process is not correctly carried out, there are tons of health issues that can result from it. I'm going to highlight just two of these health issues, which I think everybody would be common with, right? On the next slide. But don't forget, like I said, What I'm trying to understand is how exactly do we go from having just one egg and just one sperm to a full human? How exactly are we able to help these cells stick together? And so in the lab, every day I go into the lab, I try to do a bit of work to understand better. I'm going to talk about the techniques that I'm using, right, to study cells and how cells are able to stick to each other. But like I said, if this process is not carried out correctly, there are problems that can result. And don't forget that the goal of research is to solve problems. Here we can see a person with tumor, cancers, right? When the cells do not stick to each other correctly, several disease conditions can result. Here, on the person on the left has cancer, and cancer is one of the deadliest diseases in the world. And so how exactly are we going to be able to fight and cure cancer? We need to know exactly how those cells are ordered. And so every day I go into the lab, I think about how exactly am I going to be able to help make millions of lives better, right? And so knowing that my research can help contribute to solving the cancer problem gives me motivation. Don't forget, like I said, the goal of research is to solve problems. And so cancer is a terrible disease. I even think that cancer is deadlier than HIV. It's true that HIV can't be cured, right? But it can be controlled. But cancer is difficult to control, especially when the cancer has reached a stage called stage 4 cancer. So you have 4 stages of cancer, right? You've heard of stage 4 cancer. What happens in stage 4 cancer is that cancer that starts at, let's say, the lungs, it travels all over the body and affects every part of the body. And so in my research, I'm trying to understand how cells stick together and how I am going to be able to help prevent cancer cells from spreading all over the body to cause cancer in secondary sites. And so the question now is, how exactly do I conduct my research? Don't forget, I'm going to repeat myself 100 times, right? Don't forget, the goal of research is to solve problems. So like I said earlier, you want to look around yourself. What are those tiny things you can solve, those problems around you? Do you think that there's too much disease in your area, like too much malaria? What's the number of students with malaria, right? Just try to have that problem-solving mindset, right? Please don't forget that. And then I've told you about my research, and I've told you my research, I'm trying to understand how exactly cells are able to stick to each other during growth and development, right? From the fertilization process in the womb to the full growth of from a baby to a full human, right? And I've told you that when we're able to understand how cells stick to each other, we'll be able to understand how cancer occurs. Because what happens during cancer is that cells divide, and then instead of sticking to each other, they just move apart. And that's why you have swelling seen in the tumor on the face of the patient on the screen. And so the question I'm trying to answer now is, how exactly do I investigate cell-cell attachment? There are 2 techniques I use mainly, right? I use X-ray crystallography. And what happens in X-ray crystallography is that I find those proteins that are responsible for helping cells stick to each other. I take those proteins, I make crystals of those proteins. So this is an example of a crystal, right? I make crystals of those proteins. A very common example of crystals you might have seen in your home is this common table salt. If you look closely at table salt, you will see how it's shaped. So you can get the proteins that help cells, that help humans grow. You can get them to form crystals. And when they form crystals, you can determine the structure of the protein using X-ray diffraction. And so you take your crystals, you put it in front of an X-ray source, and then the X-ray is shot through the crystals. You have, and then you're able to determine the structure of the protein using the reflections or diffraction pattern generated by the X-ray. And one of the drawbacks of this type of research is that it's really expensive, right? And so, for instance, To get to even be able to shoot your crystals, the university pays a ton of money to national labs. I think about $300,000 yearly. And so I think if you're interested in structural biology, you should begin to look at universities in the US, universities in the UK, universities in France. I think these are like powerhouses that are that carry out research in determining protein structure. And so when I know the structure of the proteins, I'm able to determine how the protein acts normally and how the protein acts in patients with cancer. And so when you know the structure of the defective protein or the structure of the faulty protein now in cancer patients, you're able to design a drug to target these proteins and to make them work better. And so that's like the long and short of what I'm doing at the university. And it's really important because there are tons and tons of diseased patients that need help, that need new treatment. And so we need to be able to make life better for people. And so this motivates me every time I go to the lab. And so I've just talked about one method that I use to carry out my research, which is X-ray crystallography. And then a second method for determining the structure of proteins is electron microscopy. And so what happens is, some of you are biochemists, right? And so I believe that you've worked with proteins, at least as part of your undergraduate curriculum. So you get these proteins, they're just like water, right? They're liquid, right? You get these proteins, you put them on slides, you take them to an electron microscope, and then you take pictures. Don't forget that what the microscope does is to enlarge small, minute specimens. And so you take your proteins, you put them on a slide, and then you take them. So this is like an electron microscope, right? And then you put them in the electron microscope, and then you take images of your protein. And with this, you're able to determine the structure of a protein. So this small image I've put here that you see with different colors is the model of a protein. So when you're done solving the protein structure, you're going to see the protein like this on your computer, right? One of the drawbacks also of this is that it's really expensive. You can get the microscope for, I think, $5 million, right? And so I think if you're interested in doing structural biology, it's important that you look at universities with the resources. You can talk to Paninga. If you have questions, please talk to Paninga. He can create a database of some of these universities and you can look through if you're interested in structural biology. For the biochemistry graduates in the house, I think structural biology and biophysical chemistry is very interesting for you if you want to pursue a career in research. I've been doing this for the past 3 years, and trust me, it's been interesting. And now to bring it home, like I said, I'm trying to understand the structure of proteins, right? And I've used these 2 different techniques. And so these techniques are not just limited to the type of research I'm doing. A very common example, which we've all seen today, right, is the coronavirus. When you see this structure, you guys have seen a lot of these type of pictures where you see how the coronavirus has the spikes or around it. The question is, how were they able to determine the structure of the coronavirus? They used an electron microscope, right? And so these techniques are very powerful techniques for understanding how proteins work. They're very powerful techniques for understanding what happens when cancer occurs. What happens to those proteins? How are they defective? How have they changed in their structure? And so I think I would not go in depth because I don't want to make this technical, right? But what I want to just bring to your knowledge is that structural biology is fantastic, and it's a very interesting way to understand the structure of molecules and how these molecules might cause diseases in humans. In my case, I'm looking especially at cancers, right? And so, but you can just make up your mind. You might decide to study other diseases and how all these macromolecules cause these diseases. And so the last question I will try to answer is how exactly do you get into structural biology? Because structural biology is resource intensive. You might likely need to go into graduate school. You might likely need to do a PhD, right? And so I've just shown some of the things you need here. First, you need to look at graduate programs. For American universities, most of these universities, they start accepting applications in September. So the first thing I would like you guys to do is to build a list of universities that you might be interested in and that offer programs in structural biology and biophysical chemistry, right? The next thing you would also want to do is that while applying to graduate school, while applying to PhD programs, you will need letter of recommendations. For those of you in second year undergraduate university, undergraduate of your undergraduate degree, for those of you in third year, fourth year, one thing you need to start doing now is to start getting close to your lecturers. Because when time to apply for your PhD comes, they will need to write recommendation letters for you to say, oh, I know this student. I think he's intelligent. He has done this in the past, and I think he would succeed in your graduate. So please start getting close to your lecturers, to your professors, even the graduate teaching assistants that you have in your university right now. Please start getting close to them, right? You would also need a CV to apply to graduate school, right? And there are so many online samples that you can use to create a CV. You would also need to write the personal statement while applying to graduate school. You can always talk to the chief steward, Paninga Mulia. He can always provide resources. You can email me. I'll be willing to provide resources if you need to apply to graduate school. You might need to write tests to test your English language proficiency. And so you will also need to look at funding options. However, for most PhD programs in the US, most PhD programs in the Western world, the US, Europe, scholarships come with your admission, right? So if you're able to get admission into a university, you most likely get a scholarship to go with it, right? So for you to be able to study structural biology, the easiest part is to apply to university and to do a graduate program in the university. And so I hope that over the past few minutes, I didn't want to take too much of your time because Paning has warned me that I have to be on time, right? Is to apply to graduate school. And so I'm going to stop here and I'm going to take your questions. And if there are other things that you think you're interested in asking, please ask those questions and I'll be willing to provide an answer. And so on the next slide, I put a bit of a dad joke. I hope you find it funny. So please, if you have questions, you can ask now. I'm done with my presentation. I hope I didn't take too much time, right? And so please ask your questions.
Thank you so much, Divine. It was a thoughtful session because I was thinking, okay, Bamalaga is a student. I just graduated last month, and I wish I had this information at my fingertips. But thank you. I know it's not late. It's not. Thank you so much for the privilege to actually listen to you. So I was actually going to ask why the session was going on. I was going to ask why you became interested in the particular structural biology, how you became interested in it. But I think you answered that later on. But then when your bio was being read out, I didn't take note of you doing a master's program.
Oh no, I don't have a master's degree.
Okay, so how did that happen? I thought it was like a step-by-step process, like gradually the master. So I think I need to be enlightened on that. While we wait for other people, please leave your questions in the Q&A section, or you can use the chat box. Please drop your message there. Thank you.
Okay. We'll come to you, Timisola. But for most universities in the US, you don't need the master's degree to apply for a PhD. So once you're done with your undergraduate degree, you can apply directly and come for a PhD. So I don't have a master's degree. So it's for universities in Europe, you need a master's, right? But for universities in the US, you don't need the master's. But the PhD is a bit longer. It's like 5 years, right? And so during that 5 years, they expect you to catch up and then understand a lot in your field. So if you don't have a master's degree, it's fine. You can apply to doctoral programs in the US. Yeah, Timmy Solar.
Hello.
Yeah.
Good evening. Yes, I want to ask, I'm an undergraduate, level.
Okay.
Studying medical biochemistry. Okay. I wanted to ask, can I engage in research at this level?
I think you're asking this question at a very good time.
Yes. Because I really want to engage in research. So as my earlier stage. So I want to ask, how can I go about it?
So one thing I did when I was an undergraduate was that I went to a lecturer, right? And then I told him that I wanted to work with him, right? And so what I did was I volunteered to work for a professor. And so, professors have doctoral students, right, that do research. So I was just working with the students and I was gaining research experience. So I think one thing undergraduates need to do is that go talk to your lecturers. Don't be afraid. They want you to come to them. You can work with their PhD students, with their master students. You can learn how to do research a little by it. And then it helps you. The experience is fantastic, And so I think one thing you should do is to just volunteer to work with a lecturer right now. Work with his masters or his PhD students. You'll be able to do lab research with them right now. So I think that's something you should think of. So you might not be able to do the research by yourself, but working with a lecturer, volunteering with a lecturer is the right way to go.
I would like to ask a question, Divine.
Okay.
So thank you very much for that brilliant presentation. It's really glad to have you on the call. So like Divine was saying earlier, the whole package, resources to make available, that's going to be available from the mentorship aspect of this program. So while the program unfolds, we are going to open applications for people, because it's not going to be everybody on the call. It's going to be people who are really interested and are willing to commit themselves to the process. So you would have access to people like Divine, who would be willing to guide you through, while you study, and even prepare you for research on advanced levels. So, but my first question is, you said something about your research being expensive. So, and I'm wondering, because somebody asked a similar question yesterday, what can somebody who is an undergrad begin to do now on a small scale? Like, is there something one who is interested in structural biology can begin to do on a small scale, right now, coming from a place like, say, Nigeria, or maybe an African country that does not necessarily have the resources to conduct such research?
And that's a very interesting question. It's a bit difficult to answer because of the resources involved, right? So structural biology is expensive. So most universities in Africa, I think just South African universities, might be able to do research in structural biology. So one thing I want people now to do is that I think if you can learn to code in Python, especially, most of the applications they use in structural biology are Python based, right? So if you can learn how to do data analysis in Python, by the time you start your actual research, your actual PhD research, it's going to help you a lot, right? Because you're going to be able to use those analysis software that you would use to create your protein structure or solve the protein structure. Once you have those Python skills, it helps you a lot, right? But in terms of the groundwork itself, enzymology in Nigeria, I know Nigerian universities are able to do they do research in enzymology, right? For those in biochemistry. It will also help you when you want to do structural biology, because at least you understand how proteins work, you understand kinetics, and so. But I think one thing you want to start to do right now is to learn how to code in Python, and to build small projects in Python. Because when you start the actual structural biology, the softwares you will use are Python based. Most of the software you will use are Python based. And so I think that's one good step to have. And like I said earlier, look for a lecturer in your university, attach yourself to his students, work with them. It would help you a lot, even if it's not directly related to proteins and structural biology. There are certain skills you need to learn, like how to make dilutions, how to grow bacterial cells, how to do certain things that can be applicable to research in structural biology. And so here we have a question from Godson Silas. He says, how do I position myself for research as an undergraduate interested in research? Like I said earlier, it's hard for you to lead your own research because you might not have as much knowledge as someone that has been in the field for like, say, 5-10 years. So the best way to start out is to start with someone. And then you can now look around yourself. For instance, I have a friend, right? Not in biochemistry, but in a related field, in economics. And so he was talking to me one day and he just said, we really don't have data on how much money is in the informal sector. And what did he do? He just printed questionnaires and started going around some local governments in Ebonyi State to like start collecting data. So let's say after 2-3 years of doing undergraduate research, I'm assuming now for someone in 100 level, you'll be able to start coming up with independent ideas on how to solve certain research problems around you. And hopefully the mentorship program would be great. You can always talk to your mentors here in graduate school. They have a lot of experience with doing research. And so they'll be able to help you get in. And so there's a question here that Anonymous, he says, what can one do when his prospective PI in the US is just into elementary science, which is not application driven? How can he solve problem with such a PhD degree? This is a very good question because this person has a lot of knowledge. So the truth is that there are almost like two types of research, or like three, especially in the biosciences. You have basic research, you have translational research, and then you have clinical research where you're directly working with the patient, right? So if you think that your PI is doing basic science research and you don't like it, I think the truth is that every PhD is solving a certain problem. The problem might not be in clinical research. So for instance, I'll give you an example of a clinical research problem. Let's say someone comes to the hospital with malaria, right? Which is like a common example. And then the temperature is very high. When you make a drug specifically for that patient and then you treat the patient, you can see the direct application. You can call that clinical research, right? This is a very rough example. However, when you make an example of basic science research now is that when you take the blood of the patient and try to understand how exactly the malaria is able to get a hold of the patient, you can think of it as basic science research because you might not necessarily be seeing the patient. You're not going to see the patient. You're just looking at the blood of the patient or the proteins from the patient or the macromolecules from the patient, right? So that's basic science research. So if you think you're not interested in elementary science or elementary research, then please don't do it because a PhD is five years. It's a long time to do something that you don't like. So I think the best thing you should do is to look for a research group that does something you enjoy and to join that research group. I think it's the most important thing. So you don't want to get stuck doing something that you don't like. You want to be able to go to the lab every day with smiles, knowing that you enjoy what you're doing. If this answers your questions, please let me know. If you think you still need more answers, please also let me know. Paninga, you would like to answer some of these questions?
Okay. I think I'm one person who can really relate with the question he just asked, but I wouldn't go so much into the details. So there are, of course, every research solves a problem, but then the problem may not be of interest to you, right? Problems or interest is relative. So it's best to do what, like Divine rightly said, do what is of interest to you. And so you can do it easily. While people will be doing it for work and it's going to be so exhausting, it's going to be so disturbing, you would have some fun, at least, you'll have some motivation to do it. So it may not necessarily be easier, but at least you would find some joy doing that kind of work. That's really the best thing I would say right now. I'm about to ask you one more question, Divine, by the way.
Okay.
Because while one considers research, I think one of the things people would equally be looking forward to is the prospective career options in that field of research. So as one who is doing structural biology, what are the prospective career options?
I think I should have added a slide on that. That's a very good question. And so most of the career options in structural biology are in the biopharmaceutical industries, right? And so most of these large biopharma companies, Pfizer, GSK, Merck, Johnson and Johnson, they have structural biology centers where they solve the structures of different types of viruses, different types of proteins, so that when they make new drugs, they can test it against those proteins that they've looked at their structure. So most of the job opportunities in structural biology are in the biopharma industry. Also, they're national labs, right? They're like large laboratories in the US, in Europe. There are labs in Nigeria. There's not much structural biology work going on in Nigeria, right? But they're like large labs all over the world where they employ structural biologists to help solve problems. So yesterday I was just looking through Twitter, and I looked at Frederick National Cancer Lab here in the US, and currently they're employing a structural biologist, right? They just put up this job vacancy yesterday, right? And so they are looking for someone that can be able to help solve cancerous proteins, structure of cancerous proteins using electron microscopy. And so most of the job opportunities are in the biopharma industry, and there are opportunities, a lot of them. So when you're done with your PhD, you would move on to so working as a structural biologist in maybe Pfizer, maybe Gilead Sciences, maybe GSK, maybe there are tons of those biopharma companies in the US, in Europe, a few in South Africa. So there are tons of opportunities for structural biology. So it's not something, now when you get into school the first time, I remember those days when I was in my first year. And then I was asking, I say, waiting with use this thing study. And then they will tell us, you can work anywhere. Don't worry, this one is not like that. It's not about you can work anywhere. There are specified jobs that you can take, and then there are specified career openings for you. So if you do structural biology, you should be fine. You'll get a good job, and you would have money, so you'll be fine. And so there's a question here from Timi Sola DBC. It's, please, how can I reach you if I have some questions to ask, right? Paninga, please correct me if I'm wrong. You intend to when the mentorship stuff works, you're going to give them our contacts, right? In case they want to reach me later.
Yes, we will.
So please feel free. You can just send me an email. I'll reply as soon as possible. And we can keep talking from there.
Okay. Thank you so much, Divine.
Thank you.
It has been having you on the call. I think this is one of the things that we are grateful to Paninga, to Pani World Foundation, because at zero cost we are getting this information. At zero cost we are having access to people on a normal day we are not going to meet walking on the streets. So thank you so much, Paninga. And with this, I think we have come to the end of today's session. Please follow Pani World Foundation on social media, on Facebook, on Twitter, on YouTube. Yes, subscribe to our YouTube channel. This is where the various recordings from the summer program is going to be sent to at the end of the 6 weeks summer program. So I think, thank you so much. And we're going to be meeting tomorrow. No, we're going to be meeting next weekend by 3 p.m. W8 EST and 8 p.m. WAT. Thank you so much, guys. I really want to see us all next week. I think I saw some familiar names that joined us yesterday, and new persons, participants that joined us today. I know it has been an enriching section. And I think one of the things, the last slide he shared was how to get into this program to check as students can check for graduate programs, then get close to a lecturer, professor, and see how you can assist. Volunteerism has actually given a lot of people an edge in applying for these applications and sending out applications. So you could get close to your professor, your lecturers, and see how they can even put you through at some of these instances. Then prepare your resume and CV. You're going to be writing your personal statements, take admission tests when need be, and you get funding options. And for some, funding comes with acceptance when you're accepted into the program. So thank you all once again, and have a good night rest for those that in the time zone where it is night, and for others can continue to have a lovely day. Paninga, I don't know if you have anything else to say before we end the call.
No, that's going to be all from my end. Thank you very much for joining the call.
All right. So we'll see you all next week. Please make it a date. Thank you.
All right. Bye, everyone.